Hi everyone, I'm Ronaldo Nunes. I'm a Brazilian PhD student at UFBC in São Paulo, Brazil, in the program of Neuroscience and Cognition. And from June to August, I participate as a student in GSoC. So the title of my project is Conversion of Large-Scale Cortical Models in PyNN and NeuroML. Just to explain it a bit, in this project I propose to convert two published large-scale cortical models, one for the Maca Cortex and other for the human cortex to PyNN and NeuroML. Large-scale cortical models simulate the behavior of interacting cortical regions. In these models, each cortical region is simulated using a spiked neural network or firing rate models, and long-range connections are given by anatomical data, usually obtained by tracer injections or diffusion tensor imaging, DTI. The idea to convert to PyNN and NeuroML is because both are frameworks for the simulation of neural networks and they are independent of a simulator. So a code written for PyNN or NeuroML can be run using the backends of popular simulators such as Neuron, Brian, and Nest. So I think that the best way to get familiarized with the project is through this report. That's the last report that I've made for the project, where I summarize what I've done, the results that I got, and the challenge that I've got during the project. So here we have links to the papers and links to the codes here, and for the second part of the project here, right? I dedicate most of the time for the first model, the large-scale cortical model for the Maca Cortex. This model was developed by Joe and colleagues to investigate the propagation of activity between cortical areas. Here we have the reproduction of some figures from the paper using codes that I've implemented during the project. It was possible to reproduce most of the figures because the authors provided the original code on GitHub. I'm not going into the details for this model, but the idea is to show that increase the weight for long-range excitatory connections and local inhibitory connections, it's going to cause an enhancement in signal propagation among cortical areas. So it's clear by this figure. Here, for weak connections, we have some propagation between cortical areas, showed by this raster plot. But when we make this connection stronger, this propagation increases. And it happens for asynchronous stimulation and for synchronous stimulation as well. For the second model, we only started the conversion, but here there are some figures for the anatomical data, for the human cortex, and the state variables in the model. Currents, firing rates, as variables that they are the synaptic gating, and the simulated bolt signal. So for each model, we created a GitHub repository, and they can be accessed by these links. So let's take a look at the codes for model 1. So here we have the paper's reference and the link to the paper. Codes are separated by programming language and the figure that they can generate. Here we have the original MATLAB code and the original Brian code, and as you probably know, MATLAB is paid, so I converted all the MATLAB codes to Python, so they are free available and they can run in Python now. But also I have created optimized Brian codes, and of course codes in Pine that also Python but using the package Pine and NeuroML. So let's take a look at the Pine repository. So here in Pine repository, it's still a work in progress. There's some work that should be done, but you have the command where how you can run these codes, and the arguments required, the explanation for each argument, and here are some examples. So if you run a command like this, you're going to generate this figure, and so on. So here Pine runs using the backend of Neuro or Nest, you can choose. And for Brian 2 it's not yet available because Brian 2 is not available in Pine yet. But I think that it's a work in progress, right? So for those who are familiarized with computational neuroscience, the synapses can be simulated using delta function, alpha function, exponential, or double exponential function, also called bi-exponential. In the original code, in the original Brian code, the synapses were simulated using delta function, but these kind of functions are not available in Pine. So to overcome this problem, I figured out how to make an alpha function behave like a delta function. Basically, to make it short, I decreased the time constant and I found a factor to make the peaks similar. But here, in this link, Jupyter Notebook, I show all the steps to go from a voltage-based delta function to a current-based alpha function, preserve the dynamical behavior. For example, here in this Jupyter Notebook comparison, you can see a comparison between a membrane mutation, a peak membrane mutation generated by a delta synapse and an alpha synapse, right? And they are quite similar. It's for excitatory synapses, excitatory neuron, and here for inhibitor neuron. Right. So now let's take a look at the model 2. So here's the repository for model 2. Here's the paper's reference and the link to the paper. And if you go here in the Python folder, you're gonna see that it requires a package developed by the authors of the paper. They developed a package to run the simulation, and what I did, I created a Python code here in the folder large scale to generate figures for the state variables and the anatomical data. Here's the command, here's the argument, and here there are some examples of commands. Anatomical data and state variables as I showed in the report. Right? Well, I hope you have enjoyed this walkthrough. I'd like to say that this project was possible because the support from INCF and my mentors, Padraig and Ankur. And of course, if you have any questions, this is my email and this is my Twitter. Thank you very much.